Hello, everybody. Today we're going to continue our lecture on replication of cytoplasmic large DNA viruses. Poxviruses can be used as an example. These lecture notes can be found as part of lecture package number 12, large DNA viruses. Slide 34. The family Poxviridae are viruses that can infect vertebrates and insects. These viruses are large and they are the shape of a brick, as is depicted on the upper right hand side picture. It has an external and inner envelope. It has lateral bodies and a complex coat of tubular structures. The very unique issue about Poxviridae family is that their replication occurs in the cytoplasm, as opposed to most of the viruses that we have studied so far, where the replication occurs in host cell nucleus. Poxviridae family of viruses are capable of causing benign tumors in experimental hosts as well. Slide number 35 is basically a depiction of the vaccinia virus variant that we just basically talked about. Slide 36. Human pox viruses cause characteristic rash and pox. Viruses in this family that we'll talk about are variola, vaccinia, and molluscum contagiosum. Variola causes smallpox. The virus is transmitted by inhalation and infects respiratory tract cells. From there, it causes systemic infection. The good thing about variola is that World Health Organization initiated a vaccination program, and the disease was eradicated in 1977, about over 50 years ago. The second virus in the pox viruses family is vaccinia, that causes cowpox in the laboratory settings, and the virus can be used as the recombinant vaccinia virus can be used for vaccination purposes. Third virus is molluscum contagiosum, that causes localized lesions and is being transmitted by contact. Even though pox viruses are mainly confined to human hosts, but sometimes we see occasional pox virus zoonosis that to humans, that these are diseases that will usually cause localized lesions and can be transmitted by contact. In sheep and goat, we see a disease called ORF. In rodents, cats, and cows, cowpox, and in monkeys and squirrels, we have monkeypox. Vaccinia virus genome is a double-stranded DNA virus, linear, about 186 kilobase per, relatively large. It has covalently closed ends that forms hairpin loops. At the ends of the genome, we have so-called IR, or inverted repeats. Since it's at the end, we call them inverted terminal repeats. In the middle of the genome, or center of the genome, we have a unique sequence or conserved central region about 160,000 base per long, that the genes code for enzymes that are required for DNA or RNA synthesis. Slide number 39 is a definition of tandem repeats and inverted repeats. Tandem repeats usually happen in DNA sequences where a pattern of one or a few nucleotides are being repeated, and these repeats are directly next to each other. Whereas an inverted repeat, or IR, is a single strand sequence of nucleotides that is followed by downstream reverse complement of its sequence. Reverse complement, meaning that it's the opposite direction, and the sequence of the nucleotides is complementary to the original strand. I would strongly recommend you guys to take a few minutes and read through these definitions, even though it's assumed that you've had them in prerequisite courses. So vaccinia virus, let's talk about its entry and uncoding. Vaccinia virus's entry can be either via fusion of the virus with host cell plasma membrane, or it can bind to specific receptors on the host cell surface and enter through receptor-mediated endocytosis. Vaccinia virus, once it gets into the cytoplasm, the viral core will be released, and virus has these proteins shut off host function. So once they come in, the virions or viruses come in, they shut off host functions. Therefore, host is at the service of the vaccinia virus. And further encoding of the virus leads to early viral gene transcription and protein translation that occur in host cell cytoplasm. So in terms of the expression of vaccinia virus early genes, virus core brings in the enzymes that are needed for the transcription of virus. Half of its genome is expressed from early gene promoters. And we've talked about this issue before when we touched on gene expression, that a promoter won't get turned on or activated by itself. It requires a bunch of proteins that can bind to DNA, so-called DNA binding proteins. These can be activators, transcription factors, and I don't want you to go through the details of it, but chromatin remodeling enzymes and proteins, etc. It's not a simple process. It's very complicated. But the net result of early gene expression is expression of those enzymes that are required for viral DNA replication. Slide number 42, how does the vaccinia virus DNA replication happen? We talked about the fact that replication of this virus occur in cytoplasm. Viral enzymes recognize and replicate those circular DNA molecules at the end of these circular DNA molecules, virus causes a nick, and it creates a free hydroxyl group, lead to self-priming, and this self-priming triggers the synthesis of DNA on one parental strand. Remember, we said that vaccinia virus is double-stranded DNA, so it occurs on one parent strand. And continued DNA synthesis forms a concatemer. That the concatemer is later on cleaved into genome size nucleotide sequences. And if there is mixed infection, presence of mixed infection results in higher rate of recombination between viruses. So imagine if you have two viruses at the same time, and there is a recombination event happening when the virus uncoats and DNA is ready for replication and unwinds, results in new genome structure and leads to new viruses. What about vaccinia virus expression of late genes? Slide number 43. We have early gene expression, DNA synthesis, then there would be a switch, that this switch is due to viral regulatory proteins, as well as the configuration of newly replicated DNA molecules. So these two events lead to a switch from early gene expression to late gene expression. These two events are one, presence and action of those regulatory proteins that belong to virus and they come in with virus, as well as shape or configuration of newly replicated viral DNA. These result in the activation of late promoters and expression of late genes and then later on translation of these late genes, resulting in the expression of some enzymes and mainly structural proteins for packaging the virus and assembling the virus. In terms of the assembly and release of the virus, we've talked about this issue before, that in some instances it doesn't happen in one stage. It happens during sequential developmental stages, meaning that specific protein, and protein DNA interactions need to take place in order for the genome of the virus and capsid proteins to interact and assemble. These events happen again in the cytoplasm of the host cell. And viral membrane forms crescent and circular structures. there would be a nucleoprotein mass, combination of nucleic acid and proteins, that this mass forms with immature envelope and then buds through Golgi membrane for later on the release and formation of an enveloped virus. So nucleoprotein is being made, goes through Golgi apparatus, modifies, goes to a specific region of the plasma membrane. Then the virus is targeted to exit through that host cell plasma membrane that's being modified by virus. And virus is being released by budding through plasma membrane. So vaccinia virus is an enveloped virus. The replication cycle of vaccinia virus, what we just talked about, is depicted in slide number 45, and I would strongly recommend you guys to take a look at it in case there is not enough clarity in my explanations. Slide number 46. The unique feature of pox viruses among all other DNA viruses is the fact that they replicate in the cytoplasm of the host cell, not in the nucleus of the host cell. So therefore, since it's outside of the nucleus, it needs to bring in all of the various enzymes, transcription factors, everything required for its replication. Therefore, it needs to have a larger genome size. Slide number 47, smallpox virus. Is it something that can be considered as a potential terrorist weapon? Okay, so let's talk about a few issues, then we can decide whether it can be considered as a terrorist weapon or not. There's always susceptible population. The virus can be easily transmitted via inhalation. And there are some strains of virus that are highly virulent, meaning that they have high mortality rates, highly virulent, meaning that they are pathogenic. They can cause disease easily, and they lead to the death of the infected individuals up to 40%. So all of these make the virus as a potential weapon. So therefore we have all the right to be afraid of it. The issue is that smallpox virus is only stored in 2 public health laboratories, and these are located in the United States and in former Soviet Union. Slide number 48, summary, pox virus. Pox virus is a complex virus that has very large DNA genome because it replicates in the cytoplasm of the host cell. Therefore, it needs to bring in all the enzymes necessary for its replication. It has a unique structure. Remember, we said inverted repeats and has circular ends, and we have a conserved central region. And the replication strategy. Replication happens in the host cell cytoplasm, not the nucleus. For the replication of the virus, we need to have viral enzymes that are required for DNA and RNA synthesis. Smallpox has been eliminated in the natural environment. It is debated whether we need to fear it as a potential terrorist weapon. If so, then what would be the best defense strategy? This concludes the material for lecture set number 12. The assigned reading is from chapter 18 replication of cytoplasmic DNA viruses. Thank you so much for your attention. Have a wonderful day.